Hi everyone, I'm Dr. Nishanth Ulhas Nair. I work at the National Cancer Institute at the National Institutes of Health. I'm a computational biologist by training. I'm going to discuss my work, Putative Functional Genes in Idiopathic Dilated Cardiomyopathy. This was published in Scientific Reports in 2018, just a few months back. So idiopathic dilated cardiomyopathy, or DCM, is a heart disease. It is a common form of cardiomyopathy and a major cause of heart failure. It is characterized by the enlargement and weakening of ventricles. It is a complex disease whose molecular mechanisms and key genes or causal genes are poorly understood. The research question that we would like to ask is, can we identify functional genes in idiopathic dilated cardiomyopathy? To address this question, researchers earlier have used a differential gene expression based analysis. Differential gene expression based analysis is useful, however, it cannot distinguish between functional genes and downstream effects or co-expressed genes, and therefore we need to consider additional features. Various other studies also which use animal models by knocking out individual genes in animal models and checking for their effects in cardiomyopathy are great. However, the animal models don't always translate to humans. So in our work, we looked at 213 human hearts, out of which 77 have idiopathic dilated cardiomyopathy, and the rest 136 are donor controls. This is from the Magnet database. We integrate a huge bunch of features: genetic features, epigenetic features, transcriptomic features, phenotypic features, network-based, and evolutionary features. We have more than 180 features, and we now build a new machine learning approach called Hridaya. The word Hridaya means heart in Sanskrit. It is a two-stage machine learning approach. It uses a whole bunch of features, and it identifies for every gene. It gives you the potential of that gene for being a functional gene in idiopathic dilated cardiomyopathy. It works in a supervised manner. Hridaya has two major pipelines. It has two estimators. The first estimator is called the disease functional estimator, where it estimates the probability of a gene to be a functional gene of any disease. And the second estimator is, it identifies the probability of a gene to cause dilated cardiomyopathy, given that it comes from any disease. And finally, when you multiply the two, we get the probability of, or the potential of, a gene to be functionally linked to cardiomyopathy. So now for every gene, we can have a potential score. Now we can rank the genes, and the top ranked genes are the genes that are likely to cause or likely to play a key important role or functional role in idiopathic dilated cardiomyopathy. Now we have a bunch of predicted genes which we think are functional. We would like to validate them in different ways to see how well we are doing, how well our algorithm is working. So first we see how well it does in predicting known functional genes in humans. And we look at the HPO dataset, which is a human phenotype ontology dataset, and we get around 80%, more than 80% accuracy in predicting known functional genes. Next, we go to mouse. We look at the orthologs of the important genes that we predict, the key functional genes that we predict, and we go to the IMPC mouse knockout database, where it's a large data set where individual genes are knocked out and their phenotypes are measured on mice. We look at whether these genes, knocking out these genes, do cause abnormal cardiac phenotypes in mice, and we find that the genes that we predict to be important, a lot of them do cause cardiomyopathy related phenotypes in mice. Next, we look at drugs that are known to be cardiotoxic. That is, they have cardiomyopathic side effects. They're given for some other purpose, maybe like cancer, but they have a cardiomyopathic side effect. We look at the drug targets and we see if the predicted functional genes are enriched for the targets of these drugs, and we find that is to be true. So, and we also find that the predicted functional genes are also enriched for drugs which are given for heart diseases. So these are a bunch of strong validations that we have that the predicted list of functional genes that we have using our machine learning pipeline is may be true. It may have a lot of true candidates. We also look at the differential exon usage between of many of these predicted genes in patients who have cardiomyopathy versus normal patients in an independent data set, and we do find they have a significant differential exon usage. This is important because alternative splicing has been suggested to play a key role in cardiomyopathy. So using these predicted functional genes, we also look at their potential roles, and we find that many of them are related to cardiac function, and they also regulate differentially expressed genes. I look at the SNPs, which are linked to the expression variants of these genes, and we find that to be strongly associated with cardiomyopathic risk. We now look at cell lines experiments in independent data, and we look at the expression changes of these important functional genes, and we are able to predict new novel candidate drug targets for dilated cardiomyopathy. Using the important genes that we predict, we can look at their expression data or the genetic profile, and we are able to stratify patients who have cardiomyopathy to two groups of patients. One group has a higher left ventricular end diastolic and systolic diameters. So these are important phenotypes in cardiomyopathy. Finally, to conclude, we have in this work, a machine learning method called Frisaya, which is a two-step support vector machine-based machine learning procedure. It identifies functional genes in idiopathic dilated cardiomyopathy by integrating genetic, epigenetic, transcriptomic, phenotypic, and evolutionary features. As a resource for future clinical investigations, we also predict a list of investigational drugs that, when given to a patient, may have a cardiac side effect. They are not given for cardiac related purpose, but they may have a cardiac side effect in a negative manner. This work was done with a bunch of different fantastic people. Mainly, I would like to thank my advisor who led this project, Professor Sridhar Hananhalli, and my close collaborator, Dr. Avinash Das, and also Professor Ethan Rupin. This work was done when I was in the University of Maryland, College Park. Now I'm at the NIH. So regarding my current work, I am interested in translational research and the genetic causes of complex diseases like cancer and cardiomyopathy. I'm now mostly working on cancer-related topics. So we have methods to predict drug combination response in lung cancer. We also have new methods to predict drug synergism. We also show the importance of cell migration in affecting breast cancer patient survival, and we also have a bunch of other topics that we are working on, and it's really exciting. Thank you all very much for listening, and have a great day.